Hello, today we're going to discuss the synapsis of chromosomes during meiosis. This is when two chromosomes of similar size and centromere location pair up for division into gametes. Each chromosome is made up of two sister chromatids, which are copies of the same strand of DNA. We will compare a pair of chromosomes in two different sheep, a black sheep and a white sheep. The black sheep has the following chromosomes where the letter represents a region of DNA and the dot represents a centromere. These do not match up perfectly as there is a pericentric inversion in the second one. A pericentric inversion is when a section of the chromosome, including the centromere, is removed, flipped, then reinserted into the chromosome. These two lines represent the first chromosome and have the corresponding regions of DNA in the centromere written out. When drawing the second chromosome, we will have to use an inversion loop to make the different DNA regions line up. Crossing over can occur when two chromosomes line up and swap a portion of DNA. This occurs between regions J and K. Now we can follow the lines to determine the four gametes produced by the black sheep from this cross. First, following our top line, we can write the corresponding gamete below, chromosome MLI.JKHG. This is the exact same as our first parental chromosome, so this gamete is viable, meaning it can successfully lead to healthy offspring. Next, we can follow line 2 from MLI.J to the crossover where we move down a strand to KLM. This produces chromosome MLI.JKLM, which is not viable because there are multiple copies of the genes found in regions M and L and no copies of the genes coded for in regions G and H. Following the third line, we get the chromosome MLKJ.IHG. This is another viable chromosome as it matches the second parental chromosome and will lead to the successful production of offspring. Finally, following this line, we start with GHK, then moving up a strand to J.IHG. This gamete, GHK.J.IHG, is not viable because it has a duplication of regions G and H and deletion of regions L and M. Therefore, after the cross between these two chromosomes and our black sheep, there are only two viable gametes produced. The white sheep has two following chromosomes. We can write each chromosome out like so, representing the crossover between regions J and K here. Following our top line, we get the chromosome MLI.JKHG. This is a viable gamete because it matches the parental chromosome perfectly and has a copy of every region present. Looking at line 2, we can go from MLI.J to the cross, moving down a strand to KHG. This chromosome is the same as the parental chromosome and will be viable. Following line 3, we move from MLI.J to the cross, moving up a strand to KHG. The chromosome produced is MLI.JKHG, which, like the first two, is the same as the parents and will be viable. Following our fourth line, we can see that we get the same chromosome, MLI.JKHG, meaning it will be viable as well. Therefore, after the cross, our white sheep produces four viable gametes that could all lead to offspring. When comparing the gametes produced by our black sheep to the ones produced by our white sheep, we can see that there is a difference in viable gametes from the two crosses. Fitness is the ability to survive and reproduce, and we can see that our white sheep should have a higher fitness value than our black sheep. This is because 100% of the gametes from the white sheep can produce healthy offspring, while only 50% of the gametes from black sheep can produce healthy offspring. Therefore, over time, more white sheep will be produced than black sheep, as they are better suited to reproduce and survive. Thanks for watching. Here's a list of my references.